So welcome to Chapter 15 on the autonomic nervous system. So the autonomic nervous system is going to regulate the activity of the smooth muscles, cardiac muscles, and various glands in the body. The operation of our autonomic nervous system is going to be really important in maintaining homeostasis. But this is going to depend on the continual input or flow of sensory input from various receptors and organs, as well as the efferent motor output from those same organs. So structurally, the autonomic nervous system is going to include the autonomic sensory neurons, the integrating centers in the central nervous system, as well as the autonomic motor neurons. The autonomic nervous system is also going to be operated without any kind of conscious control. So this is a non-voluntary sort of control here, or involuntary control. The autonomic nervous system is also going to be regulated by the brain. Primarily, the regulation of the ANS is going to be mainly regulated by the hypothalamus and the medulla oblongata. And these are going to receive input from the limbic systems and other regions of the cerebrum. So let's start by just going over a brief comparison between the somatic and the autonomic nervous systems, and we can kind of have a better understanding of the differences between these two. So the somatic nervous system is going to include both sensory and motor neurons, and the sensory neurons are going to be related to things like touch and pain, temperature, proprioception, which is kind of your sense of like self-position, so that you know how you're oriented in the world, like so that you know up from down kind of thing, as well as sight, hearing, taste, smell, and equilibrium or balance. And then there are also going to be motor neurons that are going to innervate the skeletal muscles. So that's our somatic nervous system. The autonomic nervous system is very different. The autonomic nervous system is going to receive input from sensory receptors that are located in organs, blood vessels, muscles, and as well as the nervous system itself. The autonomic nervous system is usually going to operate without conscious control and regulates cardiac muscle, smooth muscle, and some of the glands in the body. So it's an involuntary control. These tissues are often going to be referred to as visceral effectors, and this is because they're typically associated with the body's viscera, those internal organs. So the autonomic nervous system is also going to consist of autonomic motor neurons that are going to regulate the visceral activities by either exciting or inhibiting the ongoing activities of those effector tissues. So this is going to be things like changes in the diameter of the pupil, so like the dilation of your pupils, adjustment of the rate and the force of a heartbeat, or the constriction or dilation of blood vessels. All of these things are examples of autonomic motor responses. And unlike skeletal muscle, the tissues that are innervated by the autonomic nervous system are often going to function to some extent even if their nerve supply is damaged. So that's a big difference there. The autonomic nervous system is also going to receive sensory input from sensory neurons that are associated with Enteroreceptors. And this Enteroreceptor is basically a sensory receptor that's going to monitor the condition of internal organs. These are typically going to be located in blood vessels, in the visceral organs, in the muscles, and even in the nervous tissue. So a couple of examples of Enteroreceptors are going to be the chemoreceptors that are going to monitor CO2 levels in the blood, mechanoreceptors that are going to be there to sort of detect any kind of stretch in the walls of blood vessels or organs. Those are all examples of Enteroreceptors. It's really also important to remember that most of the time, with these Enteroreceptors, the sensory signals are not going to be consciously perceived most of the time. So it's not like smelling some sort of perfume or seeing an image where you're consciously aware of that stimuli. most of the time we are unaware of or not consciously perceiving these stimuli from these interoceptors. However, if you have a very intense activation of those interoceptors, it can produce some conscious sensations. So like if you were to have a crazy high spike in blood pressure, you would become consciously aware of that, but you wouldn't be consciously aware of the stretching of those blood vessels, like the overstretching of those blood vessels. It would be something a little bit different, but you'd have some sort of conscious awareness that something was off. So it's a little bit different. So, again, looking at the comparison between somatic and autonomic nervous systems. Here we have an image of just a basic somatic nervous system, our little motor unit here. So in this instance, in the somatic nervous system, we have an axon of a single myelinated somatic motor neuron, all right, that's going to extend from our central nervous system. In this case, this is our spinal cord. It's extending from our central nervous system to the skeletal muscle fiber that it innervates, all right. This is our effector tissue. This is a skeletal muscle, and our neurotransmitter for this is going to be acetylcholine. All right, so it's pretty straightforward and simple for the somatic nervous system, all right. On the other hand, the autonomic motor pathway is going to look a little bit different. It's going to consist of two motor neurons in a series. So we're going to have our preganglionic neuron and our postganglionic neuron. So a preganglionic neuron is going to have its cell body in the central nervous system, and then the axon is going to extend to an autonomic ganglion. Then a postganglionic neuron is going to have its unmyelinated axon extending from the ganglion to that effector. All right, so they're kind of going to work together in unison, or in a series. So, just looking here, there's a couple of different options for how this can be set up. So most autonomic motor pathways are going to consist of two motor neurons in series, like we said. The first neuron, our preganglionic neuron, is going to have its cell body in our central nervous system, and it's going to have a myelinated axon that's going to extend from the central nervous system to our autonomic ganglion, all right, which we can see here. So this is, and just a brief reminder, this autonomic ganglion is basically just a collection of neural cell bodies that's in the peripheral nervous system. All right. The second body of the cell body of that second neuron, our postganglionic neuron, is also going to be in the same autonomic ganglion. So we can see that here. It's going to have an unmyelinated axon that's going to extend directly from the ganglion to that effector, whether it's a smooth muscle or cardiac muscle or some sort of gland. Okay, so that's what we can see here. All right. So then the other way that we can set up our autonomic motor pathways is by having our first motor neuron extend to a series of specialized cells called chromaffin cells, and these are located in the adrenal glands, specifically in the adrenal medulla, which is the kind of inner portion of that adrenal gland. And so they're gonna that first motor neuron is going to extend to those chromaffin cells instead of our autonomic ganglion, like we've seen in the previous slide. These chromaffin cells are then going to secrete the neurotransmitters epinephrine and norepinephrine, and these will be carried through the bloodstream to whatever effector tissue is needed. It needs to be changed, essentially, and so it's the release of these neurotransmitters, epinephrine and norepinephrine, from these chromaffin cells that's going to elicit that change in the effector tissue. All right. So another big difference between the autonomic and somatic motor pathways or motor neurons is going to be the neurotransmitters that they release. So all somatic motor neurons are going to only release acetylcholine. That's their one and only neurotransmitter. However, in the autonomic motor neurons, they can release either acetylcholine or norepinephrine, right? So that's an important difference there, is what they release. Okay. So We've mentioned this in previous lectures, but it's important to remember that the efferent, or the output part of the autonomic nervous system, can be divided into two main divisions. All right, we've got our sympathetic and our parasympathetic nervous system. All right, the sympathetic nervous system is usually referred to or called the fight or flight division. And this is because the stimulation of this division is going to lead to an increase in alertness and metabolism so that you can be ready for some sort of emergency. So if someone were to break into your house in the middle of the night, your sympathetic nervous system would kick into gear so that you're either ready to fight them or run away from them. Your heart's pounding, you're full of adrenaline, you're ready to either fight them off or run away. On the other hand, we have our parasympathetic nervous system. This is called the rest and digest division. And its stimulation is going to result in the slowing down of most of the body's activities. So this is when things are chill and you're all good, and you can dedicate your cellular energy to digesting that sandwich that you just eat. Maybe have a nap. This is the rest and digest. This is the kind of the opposite side of the fight or flight. So sympathetic nervous system, fight or flight; parasympathetic nervous system, rest and digest. The other division or part of the autonomic nervous system is the enteric nervous system. All right, and this is going to consist of millions of neurons in something called a plexus, just a big kind of cluster group of neurons that's going to extend most of the length of our gastrointestinal tract. So the enteric nervous system is going to operate in an involuntary manner. It's just going to do its own thing. And although the neurons of our enteric system are going to function autonomously, they can also be regulated by other divisions of the autonomic nervous system. So like I mentioned, when we're in rest and digest mode, we can have the stimulation of the parasympathetic nervous system, which is going to really kind of egg on or encourage, also is additionally going to stimulate our enteric nervous system to digest that breakfast sandwich that we just ate. So they can work in conjunction with each other. Additionally, if somebody breaks into your house, your body is not going to waste energy trying to digest your food when you need it to get up and run away or fight or flight. All right, so although the enteric nervous system does function autonomously, it can be influenced by our sympathetic and parasympathetic nervous system divisions. So here we have just a handy table comparing the somatic and autonomic nervous systems. So that's there for your studying delights whenever you're ready to use it. So let's look at the actual anatomy of our autonomic motor neurons, or motor pathways, excuse me. So there are two types of autonomic ganglia, and remember that a ganglia is just a collection of neuronal cell bodies that's in the peripheral nervous system. So we have our sympathetic and parasympathetic autonomic ganglia. And then we could break that down even further, and there are two major groups of sympathetic ganglia. So we have our sympathetic trunk ganglia, and this is going to lie in a vertical row on either side of the vertebral column, or we can have the prevertebral ganglia, and this is going to lie anterior to the vertebral column, and it's going to be close to those really large abdominal arteries. And we'll look at some examples of each of these in just a minute. So, like we mentioned, each division of our autonomic nervous system has two motor neurons, right? The preganglionic and postganglionic neurons. So in the preganglionic neuron, they are going to have cell bodies in the brain or the spinal cord. And then in the postganglionic neuron, this is going to have its cell body and dendrites located in an autonomic ganglion, where it's going to synapse with a preganglionic axon. So let's look then at some of these more specifically, starting with the parasympathetic autonomic motor pathways. So here we have the parasympathetic autonomic motor pathway. The preganglionic axons of our parasympathetic division are going to synapse with the postganglionic neurons in terminal ganglia. Okay, most of these ganglia are going to be located quite close to the actual, or actually within the wall of whatever visceral organ that they're innervating. So because the terminal ganglia are located either close to or within the wall of these visceral organs, the parasympathetic preganglionic axons are going to be very long, in contrast to those parasympathetic postganglionic axons, which are very short. So here you can see, got in solid red are preganglionic axons, and then if you look closely, you can see are in the dotted line there, you can see our parasympathetic postganglionic axons that are much shorter because those terminal ganglia are located very close to whatever it is that they are innervating. So very different lengths there. These cell bodies of the parasympathetic ganglionic neurons are going to be located in nuclei in the brainstem and in the lateral gray horn of the second through the fourth sacral segments of the spinal cord. So remembering that a nuclei is basically like a ganglia. It's just a collection of cell bodies, but it's found in the central nervous system. All right. There is then also, so we have then two different outflows from this parasympathetic sympathetic outflows there. So we have our cranial parasympathetic outflow that's going to extend from the brain stem in 4 of the cranial nerves. And then we have our sacral parasympathetic outflow, and this is going to extend from the 2nd through the 4th sacral spinal nerve. Okay, so looking at that a little bit more closely, the cell bodies of those parasympathetic preganglionic neurons, like we said, are found in the nuclei of the brainstem and the lateral gray horn of the lateral gray matter, of the 2nd through the 4th sacral segments of the spinal cord. So you can see that here. We've got our brainstem and our sacral segment there in the spinal cord. Their axons are going to emerge as part of cranial nerves that you can see here, or as part of the anterior root of the spinal nerves down here. So our cranial parasympathetic outflow is going to consist of preganglionic axons that extend from the brainstem in 4 cranial nerves. All right, we've got our oculomotor nerve, our facial nerve, our glossopharyngeal nerve, and then the vagus nerve are the 4 cranial nerves that are going to serve as that cranial parasympathetic outflow. And then our sacral parasympathetic outflow is going to consist of preganglionic axons in the anterior roots of the 2nd through the 4th sacral spinal nerves. You can see those down here. The preganglionic axons of both the cranial and the sacral outflow are going to end in terminal ganglia, and then they're going to synapse with our postganglionic neurons. All right. So, looking a little bit more closely at the cranial parasympathetic outflow, this is going to have 4 pairs of ganglia, and then the ganglia that are associated with the vagus nerve. The vagus nerve is kind of a special one. So, the 4 pairs of cranial parasympathetic ganglia are going to innervate structures that are in the head and are located close to the organs that they innervate. So our first one is the ciliary ganglia. This is a preganglionic axon that's going to pass with the oculomotor nerve to the ciliary ganglia, and this is going to innervate that postganglionic axon from this ganglia is going to innervate the smooth muscle fibers of the eyeball. So kind of helping you move those eyes, or contract and expand the pupil. Then we have our, I always mess this one up, Pterygopalatine ganglia. It's a tough word to say. This is going to receive ganglionic axons from the facial nerve, and they're going to send postganglionic axons to the nasal mucosa, the palate, the pharynx, and the lacrimal glands, or the tear ducts. And then our third parasympathetic ganglia from the cranium is our submandibular ganglia, and this is going to be found near the ducts of the submandibular salivary gland, so kind of under the jaw there. These are going to receive preganglionic axons from the facial nerves, and they send postganglionic axons to the submandibular and sublingual salivary glands, so the salivary glands that are under the jaw and under the tongue. And then our last of the cranial parasympathetic ganglia is the otic ganglia. And these are going to be situated just below each of the foramen ova, and they are going to receive preganglionic axons from the glossopharyngeal nerves, and then they send postganglionic axons to the parotid salivary glands. All right. So then let's look at the sacral parasympathetic outflow. This is going to consist of preganglionic axons from the anterior root of the 2nd through the 4th sacral spinal nerve, so down towards the end there, towards the end of the spinal cord. And as those preganglionic axons are going to move through the sacral spinal nerve, they're going to branch off of these nerves to form the pelvic Splenkinic Nerves. And so these nerves are going to synapse with parasympathetic postganglionic neurons. They're located in terminal ganglia in the walls of the various innervated viscera whatever it is that they innervate. So from the terminal ganglia then, our parasympathetic postganglionic axons are going to innervate smooth muscle, as well as the glands in the walls of the colon, the uterus, the urinary bladder, as well as the reproductive organs. So that's all controlled by our pelvic Splenkinic nerves. All right. So now let's move on to the sympathetic autonomic motor pathways. In the sympathetic autonomic motor pathway, the sympathetic ganglia are going to be the site of synapse between the sympathetic preganglionic and postganglionic neurons. There are going to be two main types of sympathetic ganglia. We've got the sympathetic trunk ganglia and then the prevertebral ganglia. The sympathetic trunk ganglia is going to lie in a vertical row on either side of the vertebral column. You can kind of see that here in this little image. And these ganglia are going to extend from the base of the skull all the way down to the coccyx. And then the postganglionic axons from the sympathetic trunk ganglia are primarily going to innervate organs that are found above the diaphragm. This is things like the head, the shoulders, the neck, the heart, things that are above the diaphragm. All right. So looking a little bit more closely at this, the cell bodies of the sympathetic preganglionic neurons are going to be part of the lateral gray horns of all thoracic segments, and then of the first two lumbar segments of the spinal cord. And the paired sympathetic trunk ganglia are going to be anterior and lateral to the vertebral column. After the axons of the sympathetic preganglionic neurons enter the sympathetic trunk ganglia, they can connect with the postganglionic neurons in one of four ways. The first of which is pretty simple. That axon may synapse with the postganglionic neuron at the first ganglion it reaches. The second option is that an axon can move up or down or ascend or descend to a higher or lower ganglion before synapsing with the postganglionic neuron. Our third option is that an axon may continue without synapsing through the synaptic trunk ganglion to end at the prevertebral ganglion and then synapse with the postganglionic neurons there. And then our last option is that an axon can also pass without synapsing through the sympathetic trunk ganglion and then a prevertebral ganglion, and then it's going to extend to the adrenal medulla, where it would interact or synapse with those chromaffin cells. So let's look then at the pathway from the spinal cord to that sympathetic trunk ganglia. The preganglionic axons are going to leave the spinal cord along with the somatic motor neurons at the same level, same segmental level, so the same areas within the spinal cord. After exiting through the intervertebral foramina, the myelinated preganglionic sympathetic axons are going to pass into the anterior root of a spinal nerve and are going to enter a short pathway called a white ramus before they then pass to the nearest sympathetic trunk ganglia on the same side. Collectively, the white rami are going to be called the white rami communicantes, and these are going to be structures that contain sympathetic preganglionic axons that connect the anterior ramus of the spinal nerve with the ganglia of the sympathetic trunk. The white part of the white rami communicantes in the name is going to indicate that they are myelinated axons, because that myelin is going to be largely lipid and protein based. It's going to look very light in color, so it looks kind of white. So only the thoracic and the first 3 lumbar nerves are going to have these white rami communicantes. Then when we look a little bit closer at the organization of the sympathetic trunk ganglia, those paired sympathetic trunk ganglia are going to be arranged anteriorly and laterally to the vertebral column on either side. So typically there are going to be 3 cervical, 11 or 12 thoracic, and then 4 or 5 lumbar, and then 4 or 5 sacral sympathetic trunk ganglia. And then there's 1 coccygeal ganglion as well. And then the right and left side, the right and left coccygeal ganglion are going to be fused together and are going to lie along the midline, which is what you can see down here. The cervical portion of each sympathetic trunk is going to be located in the neck and is going to be then subdivided into the superior, middle, and inferior ganglia. The postganglionic neurons leaving the superior cervical ganglia are going to be responsible for serving the head and the heart. They are distributed in the sweat glands, the smooth muscles of the eye, blood vessels of the face, the lacrimal glands, so the tear ducts, as well as the pineal gland, the nasal mucosa, salivary glands, as well as the heart. And the postganglionic neurons leaving the middle cervical ganglion as well as the inferior cervical ganglion are going to innervate the heart and blood vessels of the neck, shoulders, and upper limbs. All right. The thoracic portion of each sympathetic trunk is going to lie anterior to the neck of its corresponding rib, and this region of the sympathetic trunk is going to receive most of the sympathetic preganglionic axons. The postganglionic neurons from the thoracic sympathetic trunk are going to innervate the heart, the lungs, the bronchi, and other thoracic viscera. So a lot of breathing type stuff going on there. In the skin, these neurons are also going to innervate sweat glands as well as blood vessels and the little erector pili muscles, those little muscles that are going to be attached to or wrapped around hair follicles that make your hair stand on end. So the lumbar portion of each of the sympathetic trunks is going to lie lateral to the corresponding lumbar vertebrae, and then the sacral region of the sympathetic trunk is going to lie in the pelvic cavity on the medial side of the anterior sacral foramen. So, there are several pathways from which the sympathetic trunk ganglion can get to the visceral effectors. The axons can leave the sympathetic trunk in one of 4 possible ways. They can either enter the spinal nerve, they can form a cephalic periarterial nerve, they can form a sympathetic nerve, or they can form a splenic nerve. Now let's look at each of these individual possibilities a little bit more closely. So starting off with the spinal nerve. Some of the incoming sympathetic preganglionic neurons are going to synapse with postganglionic neurons in the sympathetic trunk, either in the ganglion at the level of entry or in a ganglion that's farther up or down the sympathetic trunk. The axons of some of these postganglionic neurons are going to leave the sympathetic trunk by entering a short pathway called a gray ramus, and this is going to merge with the anterior ramus of the spinal nerve. So therefore, the gray rami communicantes are going to be structures that contain sympathetic postganglionic axons that connect the ganglia of the sympathetic trunk to the spinal nerves. The gray rami communicantes are going to outnumber the white rami because there is a gray ramus that's leading to each of the 31 pairs of spinal nerves. So the axons of the postganglionic neurons that leave the sympathetic trunk to enter that spinal nerve are going to provide sympathetic innervation to the visceral effectors in the skin of the neck, the trunk, and the limbs, including the sweat glands, the smooth muscles of the blood vessels, as well as the little erector pili muscles of the hair follicles. So we need to think about what the gray part of the gray rami communicantase is going to indicate about the axons that it contains. And so if the white rami communicantase are white because of the myelin, these gray rami communicantase, the axons that make up this pathway, are going to be unmyelinated. All right, so they're gray. Next, we have our next option for our 4 possible ways that an axon can leave the sympathetic trunk. Our second option for leaving that sympathetic trunk is going to be through a cephalic periarterial nerves. So some of the sympathetic preganglionic neurons are going to enter the sympathetic trunk and then are going to ascend to the superior cervical ganglion, where they're then going to synapse with the postganglionic neurons. The axons of some of these postganglionic neurons are going to leave the sympathetic trunk by forming cephalic periarterial nerves. And these are nerves that extend to the head by wrapping around and following the course of various arteries that pass from the head and neck, from the neck to the head. So these cephalic periarterial nerves are going to provide sympathetic innervation to visceral effectors in the skin of the face, as well as the visceral effectors of the head. So as far as the skin of the face, this is going to be things like the sweat glands, the smooth muscles of the blood vessels, as well as the erector pili muscles of the hair follicles. And then other visceral effectors of the head are going to include the smooth muscles of the eye, the lacrimal glands, the pineal glands, the nasal mucosa, as well as the salivary glands. Our 4th option for ways to leave the the sympathetic trunk is through sympathetic nerves. So some of the incoming sympathetic ganglionic neurons are going to synapse with postganglionic neurons in one or more of those ganglia of the sympathetic trunk. And then these axons of the postganglionic neurons are going to leave the trunk by forming sympathetic nerves. And these nerves are going to extend to the visceral effectors that are found in the thoracic cavity. So these sympathetic nerves are going to provide sympathetic innervation for the heart and the lungs. Our next option is the formation of a Splenkinic nerve. So some of the sympathetic preganglionic axons are going to pass through the sympathetic trunk without terminating, without stopping. Beyond the trunk, they're going to then form nerves known as a Splenkinic nerve, which are going to extend to outlying prevertebral ganglia. So our Splenkinic nerves can innervate a couple of different things. They can either go to the abdominopelvic organs. This is going to be what happens to most of the sympathetic preganglionic axons. They're going to enter that Splenkinic nerve. These are typically destined to synapse with the sympathetic postganglionic neurons in the prevertebral ganglia, and that are going to supply the organs of the abdominal pelvic cavity. The other option for these Splenkinic nerves is that they could interact with the adrenal medulla, that inner part of the adrenal gland. So some of the sympathetic preganglionic axons are going to pass through, without synapsing, through this sympathetic trunk. They're going to pass through the greater Splenkinic nerves and the celiac ganglion, and then they're going to extend to the chromaffin cells that are found in that adrenal medulla and the adrenal gland. So they do this rather than extending to another organ. When they interact with these chromaffin cells, this is going to cause the release of hormones into the blood that are then going to lead to some sort of effect elsewhere in the body. That brings us then to the autonomic plexuses, or plexuses. So in the thorax, the abdomen, the pelvis, as well as axons in both, So in the thorax, the abdomen, as well as in the pelvis, axons from both the sympathetic and parasympathetic neurons are going to form these kind of tangled network of that's called an autonomic plexus. And many of these are going to lie along very major arteries. So the autonomic plexus could potentially contain sympathetic ganglia as well as axons of autonomic neurons. The major plexuses in the thorax are going to be the cardiac plexus, which are going to supply innervations and nervous stimulations or autonomic stimulations to the heart, and then the pulmonary plexus, which is going to supply the bronchial tree. The abdomen and the pelvis are going to contain major autonomic plexuses that are often named after the artery along which they are kind of located. So these are going to include the celiac or the solar plexus along the stomach. We also have the superior mesenteric plexus, the inferior mesenteric plexus, the renal plexus, and then the hypogastric plexus. So here we have a comparison between the sympathetic and parasympathetic divisions of the autonomic nervous system. It's just a nice little table. So next let's talk about the autonomic nervous system's neurotransmitters and their associated receptors. All right. So based on the neurotransmitters that they produce and release, the autonomic neurons can be considered or grouped into either cholinergic or adrenergic neurons. Cholinergic neurons are going to release the neurotransmitter acetylcholine, cholinergic receptors are going to include nicotinic receptors and muscarinic receptors, and we'll talk about these a little bit more in depth in just a minute. Adrenergic neurons, on the other hand, are going to release norepinephrine, sometimes called noradrenaline, depending on where you are in the world. So, starting with cholinergic neurons and their receptors. These neurons are going to release the neurotransmitter acetylcholine. You can see it's in the name, cholinergic choline, acetylcholine. And these cholinergic neurons are going to include all of the sympathetic and parasympathetic preganglionic neurons, all of the parasympathetic postganglionic neurons, and then the sympathetic postganglionic neurons that innervate most of the sweat glands. So you have to take that in and process it for a minute. Go ahead. The cholinergic receptors are going to be integral membrane proteins in the postsynaptic plasma membrane, and the two types of cholinergic receptors are going to be nicotinic and muscarinic receptors. So activation of nicotinic receptors is always going to cause excitation of that postsynaptic cell. And then activation of a muscarinic receptor can cause either excitation or inhibition, just depending on the type of cell that has that actual receptor on it. So that can kind of go either way. Our other option is the adrenergic neurons, and these are neurons that release norepinephrine and are going to include most of the sympathetic postganglionic neurons. The main two types of adrenergic receptors are going to be alpha and beta receptors, and these receptors can be further broken down and classified into various subtypes. So we've got like alpha 1, alpha 2, beta 1, beta 2 receptors, based on how their structure is and what they do. So depending on the subtype, activation of the receptor can either result in excitation or inhibition. So the effects triggered by adrenergic neurons are typically going to last for a longer period of time than those that we see triggered by a cholinergic neuron. So adrenergic receptors are going to bind to both norepinephrine and epinephrine. So the norepinephrine can either be released as a neurotransmitter by that sympathetic postganglionic neuron, or it can be released as a hormone into the blood by those chromaffin cells of the adrenal medulla and then can circulate in the blood that way. Epinephrine, on the other hand, is going to be released as a hormone. So let's talk for a second about agonists and antagonists and how they relate to neurotransmitters, hormones, drugs, etc., things binding. So an agonist is going to be a substance that binds to and activates a receptor. And in doing so, it's going to mimic the effect of a natural neurotransmitter or a hormone. Right? So that's our agonist. It's mimicking what we would see in a naturally occurring neurotransmitter being bound. Our antagonist, on the other hand, is a substance that binds to and blocks that receptor, and it prevents the natural neurotransmitter or hormone from binding to that receptor, therefore preventing it from exerting that effect. So different drugs can either serve as agonists or antagonists to selectively activate or block various autonomic nervous system receptors. So a great example of this is propranolol. Right? This is often going to be prescribed for patients that have hypertension or like chronic high blood pressure. So propranolol is interesting because it is a non-selective beta blocker. This basically means that any type of beta adrenergic receptor, it's going to bind to it, and it's going to prevent their activity. It's going to prevent their activation by epinephrine and norepinephrine. So this propranolol binds non-selectively. It binds to any and all beta adrenergic receptor. All right. So the desired effect of propranolol is due to the blockage of the beta-1 receptors. This is going to lead to a decrease in heart rate as well as a force of contraction and a consequent decrease in blood pressure. However, one of the undesired effects of propranolol is going to be caused by the blockage of a beta-2 receptor. Because this is, again, is a non-specific beta blocker. It's just blocking all of the betas. So when you get this off-target effect, where you block that beta-2 receptor, this can cause hypoglycemia, which is low blood sugar. And this can result from a decreased glycogen breakdown, as well as mild bronchoconstriction, or the narrowing of the airways. So if these side effects from this propranolol are going to cause a problem for the patient, then a selective beta-1 blocker, such as metoprolol, can be used instead of propranolol. So this selective beta-1 blocker is only going to bind to that specific beta receptor, that specific beta-1 receptor. So this is a more specific and more targeted drug than the propranolol is. So this is how we look at, this is an important thing to consider when we're talking about drug design, especially. So now let's look at the integration and control of the autonomic functions. So the functions such as heart rate, the force of ventricular contraction, blood pressure, the diameter of blood vessels like vasoconstriction, vasodilation, are all going to be controlled by autonomic reflexes that are going to occur when nerve impulses pass through an autonomic reflex arc. So the reflex arc is going to be composed of a receptor, a sensory neuron, and an integrating center, as well as a motor neuron and an effector. So let's look at all of these pieces individually. The sensory receptor is going to be that's found in this autonomic reflex arc, is going to be the distal end of the sensory neuron. And this is going to respond to stimuli and produce a change that will ultimately trigger some sort of nerve impulse. These sensory receptors are mostly going to be associated with enteroreceptors. So those receptors that are going to detect changes within the visceral organs. Next is our sensory neuron, and this is going to conduct that nerve impulse from the receptor into the central nervous system. Then once we get to the central nervous system, we're going to interact with these integrating neurons, or the integrating sister. So these are interneurons within the central nervous system that are going to relay signals from the sensory neuron to the motor neurons. And the main integrating system for most of the autonomic reflexes are going to be located in the hypothalamus as well as in the brainstem. So some of those autonomic reflexes, such as those for urination, are also going to have integrating systems or centers in the spinal cord. So next then is our motor neurons. This is where the nerve impulse that's triggered by our integrating center is going to propagate out of the central nervous system along our motor neuron to its effector. And then in an autonomic reflex arc, two motor neurons are going to connect the central nervous system to that effector. We've got our preganglionic neuron that conducts motor impulses from the CNS to our autonomic ganglion, and then we have our postganglionic neuron, which conducts motor impulses from that autonomic ganglion to the effector. And then last but not least, we have our effector. And in an autonomic reflex arc, the effectors are going to be things like smooth muscles, cardiac muscles, and glands, and the reflex is going to be called an autonomic reflex. So that is the end of Chapter 15 lecture on the autonomic nervous system. If you have any questions, comments, or concerns, please feel free to email me, or we can ask during the next time we have class. So good luck.